Why abiogenesis and evolution collapse under real evidence. On the present state of physics, chemistry, information theory, mathematics, and observational biology, abiogenesis and macroevolution do not stand. Prebiotic chemistry does not reach a self-replicating, metabolizing, digitally encoded cell. Mutation plus selection does not build new body plans, new protein folds, or irreducibly complex systems within the real search limits of this universe. The numbers are not just low, they are so far beyond realistic probabilistic resources that, taken together, they function as a practical falsification of both abiogenesis and molecules to man evolution as scientific explanations of life. Abiogenesis. Prebiotic chemistry must do several things at once. It must select only one mirror form of each amino acid in sugar. It must link them into long information-rich chains in water, where such chains fall apart. It must keep them concentrated, protect them from ultraviolet destruction, and at the same time avoid free oxygen that blocks the very reactions origin of life models need. It must also generate a code and a machine that reads that code all under unguided conditions with no planning and no goal. Chirality comes first. Life uses left-handed amino acids and right-handed sugars almost exclusively. Yet standard prebiotic reactions produce racemic mixtures with equal amounts of left and right-handed forms. The origin of this holochirality remains an open problem even in current work. One wrong-handed monomer in a growing chain acts as a stop sign. As soon as a few right-handed units enter a left-handed protein, function collapses. Prebiotic scenarios must not only form polymers, they must purify them to near single-handed perfection with no intelligent selection. Next comes the polymerization problem. To build proteins and nucleic acids, chemistry must drive condensation reactions that expel water and bulk water. Equilibrium pushes the other way. Long chains tend to hydrolyze and break back into monomers. Many origin models now admit this and search for workarounds such as mineral surfaces or wet-dry cycles. Yet each fix solves one constraint and breaks another. Surface binding can hold monomers, but it can also trap chains and block folding. Wet-dry cycles require exposed surfaces, but ultraviolet light and oxidants can degrade fragile products. Now consider the stability of the parts. Ribose, the sugar in RNA, has a half-life of about 73 minutes at 100 degrees Celsius, and even at 0 degrees Celsius, lasts on the order of decades, not geological ages. Cytosine, a key nucleobase, has a half-life of about 19 days at 100 degrees Celsius and about 17,000 years in ice. These numbers look long only when compared with supposed to prebiotic timescales of hundreds of millions of years. The molecules fall apart far faster than any plausible build-up time. You do not get a growing reservoir of nucleotides, you get decay. The oxygen UV paradox adds another wall. Many proposals rely on intense ultraviolet flux at the surface to power exotic chemistry, yet significant free oxygen would generate an ozone layer that blocks the UV at the same time. Even modest oxygen levels oxidize and destroy many key prebiotic intermediates. Models that assume no oxygen gain UV but lose chemical stability. Models that include oxygen gain shielding but lack the necessary reactions. You do not get both at once in a simple, stable way. Then there's the soup itself. The classic idea holds that early oceans were rich in diverse organic matter, yet geological studies still do not provide clear evidence for such a global prebiotic soup. Modern work jumps between ponds, vents, ice, and aerosols. Each niche offers one advantage and carries several lethal drawbacks. None delivers a complete path from simple molecules to the first cell. All of this still ignores the central issue: information. A minimal self-replicating cell needs hundreds of genes and thousands of bits of specific digital information. The search space for even a modest 1 to 50 amino acid protein is about 200 novemquadrantillion, or roughly 100 quadriquinquadrantillion. Possible simple experiments at work on beta-lactamase variants suggest that only about 1 in 100 quadriquinquadrantillion Septuagentillion sequences in that space yields a stable, functional fold for a given task. Random walks through this ocean of sequences never approach even one target domain, much less an integrated network of enzymes, structural proteins, and regulatory factors. Abiogenesis needs homochirality, long polymers and water, high local concentrations, protection from radiation and oxidants, a genetic code, and a molecular machine that reads and copies the code. All in tight coordination. The joint probability for that sort of alignment under blind chemistry sinks far below Fred Hoyle's famous tornado in a junkyard 747 analogy.